Thanks for joining me again today on this trip through AJP, Heart and Circulatory Physiology, or from the editor's desk, for the May 15th, 2015 issue. This week's issue is chock full of great papers. I assure you there is something for everyone. So let's get started. To review, Matazzi et al. Discussed the many effects of CAMP kinase 2 in the heart. This ubiquitous protein participates in many cellular functions in addition to excitation-contraction coupling. The review summarizes many aspects of the molecular physiology of CAMP kinase 2 and provides a framework for understanding the role of CAMP kinase 2 in calcium homeostasis in the cardiac cell in health and disease. Lang and Kolb hypothesized that reflexcutaneous vasoconstriction to whole body cooling in older humans relies on the AT1 activation and enhanced Rho kinase activity. They studied young and old subjects using microdialysis probes that could deliver vehicle, the Rho kinase inhibitor Fasudil or losartan. The results show that AT1 receptor activation contributes to the reflex vasoconstriction in aged but not in young skin. The angiotensin component of the vasoconstriction appears to be mediated by the activation of Rho kinase. Dugal et al. Studied mutations in myosin regulatory light chain to gain insight into familial hypertrophic cardiomyopathy. They studied how disruption in actin-myosin interactions play a role in this disorder. They examined cross-bridge kinetics in contracting myofibrils from ex vivo left ventricles of transgenic mice expressing the FHC regulatory light chain mutation K104E. The K104E mutation increased the rate of cross-bridge binding to thin filaments and the rate of execution of the power stroke. Taken together, the data indicate that the mutation-induced changes in interaction of myosin with actin during the contraction-relaxation cycle may contribute to altered contractility and the development of familial hypertrophic cardiomyopathy. Finally, Tardif et al. Investigated the role of the intermediate filament protein nestin in the vascular remodeling process secondary to hypertension. Male Sprague-Dawley rats were examined 2 weeks after suprarenal aortic constriction. Carotid arteries from these rats exhibited smooth muscle hypertrophy and increased nestin protein levels. Furthermore, in cultured carotid and aortic smooth muscle cells, angiotensin II and EGF stimulated DNA and protein synthesis and increased nestin levels. These data demonstrate that vessel remodeling in hypertension may be in part due to nestin upregulation in vascular smooth muscle. The selective role of nestin in peptide growth factor stimulated DNA synthesis indicates multiple factors in the smooth muscle hypertrophic process. Thank you for joining me again on this tour through the May 15th edition of AJP Heart and Circulatory Physiology. Have a great and productive 2 weeks. We'll see you then. And don't forget, send us your best work now.